Technology. Welcome to the MOOC series, an initiative started by LG Institute of Pharmacy, Ahmedabad. In this session, we are going to discuss about the different techniques for the isolation and the preservation of microbial culture. So let's begin with our session. Microbial culture is required for growing the microorganisms in the lab. So, isolation is required for the separating a single species into a pure culture, and then that culture is to be preserved to use it for further applications. So, isolation is a process where a specific bacterial species present in a sample can be isolated. In mixed culture, more than one species are present, and in pure culture, only a single species is present. So, our aim of isolation is to obtain a pure culture of any species. There are different techniques for the isolation. The most common is the plating method. Then there can be single cell isolation by micromanipulation. There can be isolation by using physical agents like heat, pH or temperature. Or you can use different media like selective enrichment, motility media to separate a specific type of organism. The most frequently used method is the plating method, which we will discuss today. In this method, when a mixture of cells is stripped, immobilized or spreaded on a solidified agar plate, each viable cell will develop into a distinct colony. Viable cell is the living cell which can multiply and form the colony. Now what is colony? It is a macroscopic growth which is obtained from a single cell after the inoculation on the solidified plate. So the first method is trick plate method. In this method, the sample is tricked on a petri plate by using an inoculating needle or by using a nichrome wire loop. Each time before the use of the either the loop or the needle, you have to sterilize it in the flame. The loop is dipped in the culture. It is moved in the zigzag pattern on the solidified plate. Then the plate is incubated for 24 hours at 37 degrees Celsius, and after 24 hours the growth is observed. What we will observe that isolated colonies are obtained with each succeeding stroke due to the dilution of the original microbial load. Advantage is that isolated colonies can be obtained on a single plate without any dilution of the original sample. So there are different patterns of sticking. The first is a single stick where you have to start from the top and that stick have to be moved downwards up to the end of the petri plate. In triple sticking method, you can divide your petri plate into 3 zones and you can perform the sticking as shown in the figures. In quadrant streaking or multiple streaking or 4 flame method, you have to divide the petri plate into 4 sections. Then after the after the streaking in each section, in between you have to flame the loop. You will in total flame the loop for 4 times, so it is also known as 4 flame method. You have to do the streaking in one section, sterilize the loop, then from the common region you have to streak in the next section, then in the third section and then in the fourth section. Like this you have to decrease the microbial load 4 times during the process. It is one of the most important and the perfect method for the isolation of microbial cells. Second is the pour plate method. As the name suggests, here you have to mix the bacterial culture in the liquefied solid culture medium, and then you have to mix it properly by rolling the tube between the palm and pour that plate in a sterilized empty petri plate. Allow the plate to solidify, incubate it for 24 hours at 37 degrees Celsius and observe the growth of colonies on the next day. In pour plate method, first of all you have to suitably dilute the solution by up to 10 raised to 8 times. You can dilute the sample with the help of either the distilled water or the nutrient broth. The mixing is to be done at around 45 degrees Celsius to make sure that the microorganisms are living and they are not killed. You will observe that as the dilution of the sample increases, more isolated colonies are obtained on the petri plate. The advantage is that this method can also be used to count the number of colonies that is formed by the bacterial culture. The disadvantages are that it is a time consuming method. Some of the cells may get trapped below the surface which cannot be counted, and since you have to inoculate at 45 degrees Celsius, thermolabile organisms cannot be processed by this method. So this is the process which you have to perform. You have to mix, then you have to roll, you have to pour it, you have to allow it to solidify, and then you have to incubate. Okay, so as the dilution increases, you can see that the number of colonies that are formed in the plate will reduce. Spread plate method. In this technique, you have to spread the culture on the petri plate by using a sterile spreader, glass spreader. Okay, this is the best method because you will get only the surface colonies which can be counted very well. The sample is to be suitably diluted up to 10 raised to 8 times. The zero point 0.1 ml of the diluted sample is poured in the center of the petri plate and then it is spreaded with the help of glass spreader. You have to incubate the plate at for 24 hours at 37 degrees Celsius, and after that you have to observe the growth of the colonies. You will observe that as the dilution increases, more isolated colonies are obtained. What are the advantages over pour plate method? Only the surface colonies are obtained which can be counted well. This method can also be used for bacterial counting method, and you can use this method for thermolabile microorganisms also because inoculation is done in a solidified plate directly without any heating. So this is the technique. You have to take 0.1 ml from the sample suspension, you have to spread, and then you have to observe the growth of the colonies on the next day. After you have isolated the organism in the pure culture, you have to preserve that pure culture by a proper technique so that you can use that culture uh, in the subsequent practicals. So for storage, for maintaining that cells in a viable condition, to avoid any contamination of other surrounding microorganisms and to restrict the mutation, preservation of pure culture is to be done in a proper way. So the techniques for the preservation of pure culture are direct culture transfer, you can do refrigeration, you can do low temperature freezing in refrigerator or in liquid nitrogen, you can go for lyophilization method, you can use mineral oil or you can use sterile soil for preserving the pure culture. So we will see all the methods one by one. In the first method, periodically you have to transfer the culture into the fresh culture medium so that the contamination can be prevented. But there are chances of genetic mutation in this method. But still, this is one of the very easy and useful method for the performing the practicals in the lab. Second, you can use a layer of mineral oil on the top of the pure culture, which will maintain that culture in a viable state for 2 to 3 years. Frequent transfers are not needed, and the culture can be stored in a proper way like this. Next is the freezing at low temperature. Here you have to freeze the temperature in a simple freezer at around minus 20 degrees Celsius. The culture will be maintained for 1 to 2 hours, but the disadvantage is that there might be ice crystal formation or electrolyte fluctuations during the storage. Next is freezing in liquid nitrogen. Here you have to store the culture at minus 196 degrees Celsius in liquid nitrogen. Before doing that, you have to first freeze it up to minus 60 degrees Celsius for 1 hour in a normal freezer, and then you have to store it at minus 196 degrees Celsius in the liquid nitrogen. You can add the bits in the suspension. So after freezing, the suspension will be coated surrounding the bits. Then that bits can be directly used whenever you want to perform the practical. Freeze drying or lyophilization. In this technique, this is one of the best technique for the long-term preservation. Here you have to convert the culture suspension into the dry powder form by using freeze dryer. Vacuum pumps are attached, and then the liquid is converted into free flowing powder. The biggest advantages in dry form, that bacteria will be able to will remain inert for a longer period of time. Wherever required, you can reconstitute the dry powder into the broth, and you can use that broth for the practical purpose. Next is the storage in the sterile soil. In this case, this method is used for the preservation of porulating bacteria or microorganisms. You have to add the 1 ml of the suspension into the sterile soil, and then they can survive for around 70 to 80 years by using this method. This method is very useful, especially for the sporulating microorganisms. Okay, so these are the different techniques for the isolation and the preservation of microorganisms. These are the frequently asked questions from this topic, and you can have use a specific method for the specific purpose according to your requirements. Thank you for watching this video. For more such updates, like, share and subscribe our YouTube channel Pharma Ignite, and keep on watching such videos. Press the bell icon for getting the notifications. Thank you. See you soon in the next lecture.